Hi guys, so we are going to continue with chemical levels part 2. In this part, our focus will be on molecules and more specifically molecules of our lives and what basically keeps us alive. Now, because we are organic organisms, which basically means we contain carbon and hydrogen, the molecules of our body also would consider to be organic molecules. And they are broken down to four main categories of carbohydrates or carbs, lipids or fats, proteins, and nucleic acids. So for the rest of this PowerPoint, we will attempt to learn more details about each of these four main categories. Now, before I go into depth about that concept, we will learn a new vocabulary referred to as macromolecules. As the word macro implies, macro means large, so macromolecules means really large molecules. So macromolecules will be made up of individual molecules that will be connected together using covalent bonds. So I can take individual molecules and combine them together to form a macromolecule, or I can take a macromolecule and break down the chemical bonds to form individual molecules. Now, as you take a look at the diagram I have on the bottom left, you notice that you have individual molecules represented as subunits, and they have one has a hydroxyl group connected to it, and another one has a proton or a hydrogen ion connected to it. If I want to connect these two subunits together, what I need to do is I need to remove the hydrogen and the OH group as a water molecule. You can see 2 hydrogen, 1 oxygen. And that allows me to form a covalent bond between these two subunits to form a larger molecule. Now, as you understand, removal of water or loss of water is represented by the word dehydration. So if you're dehydrated, basically you lack water. And dehydration synthesis, the word synthesis implies production or creation. So basically what this reaction is telling me is that I have removed water to create a covalent bond, and that process is referred to as dehydration synthesis. Basically, I can go from molecules to macromolecules using dehydration synthesis. I can also carry this reaction in reverse, which means that I can break down macromolecules you see right here, break them down using water to separate the covalent bond. So in this case, notice you have water, it comes in, breaks down and form OH on one side and hydrogen to another subunit and causes the separation of the covalent bond that is represented right here. Now notice the word hydro. Anytime you hear the word hydro, hydro means water. So lysis means destruction. So hydrolysis means what? Addition of water to break or destroy something. In this case, your chemical bond. Now, so you need to have a really good understanding of dehydration synthesis and hydrolysis to be able to understand how you go from basic building blocks of your nutrients to more complex molecules within your body. So we're going to start by the first organic macromolecule category we have, which were your carbohydrates. Simplest way to describe the function of these carbohydrates is that they use as the main source of energy for your body. Think about glucose, because glucose is the main source of energy for your body, as you always measure blood glucose when you discuss your health or your ability to basically maintain your bodily functions. You can also store these sugar or these carbohydrates in a very small amount to use as short-term energy storage. I'm going to highlight this component for you guys. The word short-term energy storage. It is not a long-term. For long-term, we will be referring to fats or lipids to accomplish the task. Now, when we use the word carbohydrates, another way to describe carbohydrates would be the word saccharide. So the word saccharide also implies carbohydrates. The word mono, as you can see here, represents one. The word di represents two. So monosaccharides means a single molecule of sugar, and that includes glucose. As I mentioned, immediate or most essential source of energy for your cells. Disaccharides, on the other hand, is where you combine two monosaccharides via the reaction dehydration synthesis, as I mentioned earlier, the reaction dehydration synthesis, to create and combine two single molecules to form a disaccharide. And you can see here, you have glucose with the OH, glucose with a with the hydrogen group. Remove that as water, and now you have connected these two together. The example presented here is one glucose combined with a second glucose to form a disaccharide known as maltose. We can also have really complex carbohydrates that contain hundreds upon hundreds of monosaccharides. In such a case, we refer to those as polysaccharides. The word poly implies many or multiple. Now, complex carbohydrates can have two sources. They can come from your plants, as you will see on the two pictures on the right-hand side, and it can also come from animals, as you will see on the left-hand side. Notice that all of these are still made up of glucose. It's just the structures that are created, basically the branching that are created is what makes these polysaccharides different, not what they are actually made up of. So let's start with the source of animal. That is called glycogen. We have that as well in humans as well as animals. And this is basically a storage of glucose, typically in your skeletal muscle as a source of energy, or in your liver as a storage of energy. We could also have carbohydrates or polysaccharides in form of a starch that comes from plant. You feel very comfortable with that. Basically, potato, bread, pasta, rice, all of these contain a significant amount of a starch. And this is something that we can digest, we can actually consume as a source of energy. However, the second source of polysaccharides come from plants called cellulose or fiber. This is found in the cell walls of the plant. And because of the linkage, and you can see the linkage from here versus here, we do not have the ability to break down starch, and therefore we are unable to digest and use it as a source of energy. The second category of organic molecules we have are lipids or fats. If lipids are originating from plants, we refer to those as oils, and if they're originating from animals, we refer to those as fats. The most typical type of fat we have in our body is a fat that is a combination of a glycerol, which you see in green, and 3 chains of fatty acid you will see in blue. We refer to these molecules as triglyceride. As in tri implies 3, and triglyceride is referring to the backbone of triglyceride. Now, notice that when we take a look at these fatty acid chains, basically what they are made up of is carbon and hydrogen. Something else I do want you guys to notice is that I am connecting each of these fatty acid chains directly to the glycerol backbone using the dehydration synthesis. So right here is one dehydration synthesis. This is the second one, and this is the third one. I can combine this to make one triglyceride and 3 water molecules. Now, what are the functions associated with lipids? As I mentioned, it is identified as the long-term storage of energy. So basically, fat in your body is long-term energy storage, and it's very difficult to get rid of because basically your body wants to have as much as nutrients as it can, and therefore it stores it to the best of its ability as lipids. It also acts as an insulation against heat. You have a layer of fat under your skin called hypodermis, and that's basically insulates and prevents heat loss from your body, giving you the ability to maintain your 37 degrees centigrade body temperature. It protects some level of, it provides a level of cushioning around your heart and kidneys. It is found in your cell membrane, so individual cells in your body is basically wrapped with fat to set the boundary. And a lot of your hormones that are considered to be steroid hormones, including your sex hormones, testosterone and estrogen, as you can see right here, are originating from cholesterol or a derivative of cholesterol, which are also considered fat. Now, the last part of this lecture, I'm going to focus on the concept of saturated fat versus unsaturated fat versus trans fat. So first thing we're going to look at is the relationship between carbon with other carbons and the hydrogen within the fatty acid chain. So remember, fatty acid chains are basically the blue structures I'm discussing. Now, every carbon can form a maximum of 4 covalent bonds at a given time. 4 covalent bonds at a given time. So right here, if I take this carbon, if I take this carbon, I have this bond, this bond, this bond, and this covalent bond that is forming around the carbon. No double bond is present. So if there is no double bond present, and if the carbon bonds are not occupied with carbon, any remaining bond can be occupied by hydrogen. So let me repeat this. When you look at carbon, the maximum bonds you can form are 4 bonds. And in this case of the fatty acid, if the bonds are not occupied with carbon, the only other atoms they can be occupied with would be hydrogen. Therefore, we can say that the carbon is saturated with hydrogen. Because of that, we call this type of fat saturated fat. Because these have such a straight structure, they can easily pack, and therefore they will be solid at room temperature. Best example of that would be your butter or any type of fat that comes from animals. On the other hand, when we talk about unsaturated fat right here, unsaturated fat replaces the single covalent bond between the carbon and carbon with a double covalent bond. So now what you have left with is only 1 and 2 extra covalent bonds that you can form between carbon. And therefore, there are empty spaces which were potentially could have been occupied with hydrogen, and now they're not because of the presence of double bond. Therefore, we consider this type of fat unsaturated fat because they have the option, have the room or a possibility of adding hydrogen, but those hydrogen cannot be added. So, because of this structure, because of this double bond, you get kinks or knots within this straight structure. So the structure will not be as straight as you will see on a typical triglyceride, Rather, you will see kinks. These kinks prevent the molecules to basically stack up easily on top of each other, and consequently, they will be liquid at room temperature because you cannot pack as many molecules on top of each other basically. Trans fat. Trans fat is I guess it was our solution to our problems of eating fat without gaining fat. It was created by the process of heating up unsaturated fat and heating up unsaturated fat and pack it with hydrogen. Now, this is an artificial molecule that is created. It can be partially or fully hydrogenated, but the problem is that the hydrogens will end up on the wrong side of where they're supposed to be. Hence the word trans fat. Trans part means molecules are not where they're supposed to be, they end up on a different side. Therefore, because of the change in the structure, they are going to be harder to process. And they can basically clog our blood vessels and can cause major cardiovascular disease for us. We also have, when we talk about fat, we can never forget cholesterol. Good cholesterol versus bad cholesterol. Good cholesterol, as we call it in the common terminology, stands for high density lipoproteins, or HDL, which basically takes away the extra fat and transport that into your liver in a very simple term. Or you have low density lipoproteins, where it actually takes fat from your liver and transport it into your bloodstream. So when you go to your doctor and they say your LDL is high, is not a good thing because it could increase the risk of cardiovascular disease. I would strongly suggest for you to click on the two links, visit the website as well as the YouTube video to get some more information. You are responsible for the information presented on videos. Okay, I'm gonna stop it now and then we'll continue with chemical level part 3 for the next part.